have using the document camera and just checking about the tissues, a different type of tissues. And the first one, what we have started with epithelial tissue. Do you remember that last class about the epithelial tissue? Let me bring back. Okay. Go through your PowerPoints. Tissues, that's a definition which we gathered, aggregation of similar cells to perform one common function. And cell recognition by occluding the tight junction is abbreviated TJ, anchoring, adherence, and desmosomes and communication gaps. These are the tools where the cells are aggregate. We are going to see different types of cells to form different types of tissues. Okay. To start with, we have in our plate is the epithelial tissues. Okay, that the primary tissues comprises of epithelial tissue, connective tissue, muscle tissue, and nervous tissues. Okay, what I am going to discuss today is epithelial tissue and connective tissue. Okay, and that's for your exam. Okay, so please go through all these PowerPoints, and I'll come back a bit later. But I want to give an importance of this epithelial tissue. Okay. What is an epithelial tissue? Which we discussed earlier. If you see on one of the picture which we discussed last time, here it is also available in your Blackboard figures. Okay, you can also check it out. These are the derivatives of epithelium. If you see this picture, and you will see the endothelium, that lines the passageways of lymph and cardiovascular system over here. Okay. Endothelium lines in the organ cavities, that respiratory. So you can see the digestive cavity, urogenital cavity, and the epithelium covering the skin, and the mesothelium lines and closed-in cavities, pleural, pericardial, and peritoneal. So they are made up of mesothelium, and this is endothelium. This is also a type of endothelium. All of them are the derivatives of epithelial tissue. Epithelium of organs, that the kidneys and liver and pancreas and gonads. And then you can also see the epithelium function, covers the body surface and cavities, and it protects against the moisture loss and trauma, any injury, or the noxious substances. If you have exposed any other noxious substances, and these are the first line of defense of the skin. And the ectoderm, its origin is from the ectoderm, the epithelial tissue, from the embryonic development, and lines of open cavities were oral cavity, nasal, and anus. So all these parts is comprised of a specialized function as an epithelial tissue. Okay, we will see types of the cells in the epithelial tissue in a different way, like morphological types and shape of cells and different types of tissues. So the epithelial tissue is classified at different stages. Okay, they are classified as classification. Whatever I am writing it, you can write it onto your notes. Okay, and you cannot see them. 1 second, I will focus it, refocus it. I'm doing it. Yeah. Yes, now I'm just focusing, going away from it. Could you see now? Classification? Okay. Number one, squamous. S-Q-U-A-M-O-U-S. Squamous epithelium. Classification. We are dealing with epithelial tissue now today, okay? E-P-I-T-H-E-L-I-U-M. Epithelium. So the epithelium is classified into one, squamous epithelium, and two, cuboidal, three, columnar, C-O-L-U-M-N-A-R, columnar epithelial tissue. Okay. What I'm going to do, I'm going to classify depending upon the type of cells and number of layers, where it occurs, and functions of all those things. Okay. So, first one is the squamous epithelial tissue. Okay. See the morphological types. Just getting on refocusing it so that it is going to be. I'm going to do it for all these tissues. Okay, that for the squamous and cuboidal and columnar epithelial cells, all cells I'm going to classify the morphological types. How the cells looks like. Okay, the morphological, it is a simple. I'm discussing about first is the squamous epithelium. Okay, the squamous epithelium. Squamous epithelium. I put SQ so that you remember that squamous epithelium. Squamous epithelium morphological type is very simple. The simple type. Okay. And if you go on to shape of cells, the shape of cells in squamous epithelium, okay, it's like a flat plates, like it's like this. It's not it's something like, it's a very flat, these cells. It looks like this, and these are the junctions, and it's being attached to it. So the cells aggregate, that's tissue. What cells aggregate here? We are talking about the shape of cells in squamous epithelium. This is squamous. Now I'm going to get the number of layers. How many number of layers can form in this? Okay, it's going to be number of layers. Only one layer for squamous epithelium. So the squamous epithelium like this, it's only one layer. It is not multiplayer, but I'm writing a three-dimensional shape, like the cells are nucleus and nucleus and nucleus here at the middle. But it's something flat cells, but it form one layer, like a one layer of sheets, like this. This is the one layer. So this is for the squamous, for the number of layers. And the next one, it is specialization of free surface. Specializations of free surface. What do you mean by free surface of this one? The free surface is something like if you have the cells, like this, a normal cell with the nucleus and cell membrane, and this side is attached to another cell, correct? Another cell. This is called the lateral attachment. This is lateral. This is at the top. You can say the free surface. And the bottom one is a basal cell. Basal one. I'll just write it here. Could you see that one? This is lateral. That's going to be another cell. And this is the basal. This has been attached. So we are talking about now about the free surface. What is the free surface of the squamous epithelial cells? It may modify as a microvilli. Okay. Now the next one, I'll go on to the basal surface of the cell. What will be the basal surface of the cell? This is the cell, okay, nucleus, and the basal surface. This is the basal. This basal surface is modified as hemidesmosomes. Basal surface is modified as a hemidesmosomes. They are a type of cell junctions being attached to it. Okay, that's number one. We are talking about the squamous epithelium, simple squamous epithelium. Okay, simple squamous epithelium. Let's always remember that. We are dealing with it now. We are going to see the other cells as well. And the lateral surface. What is the lateral surface of this one here? Like this is the cell, nucleus. What is the modification of the lateral surface? This is the basal, but we are talking about this lateral. L for lateral. So we have done the free surface. We have done the basal surface. Now we are going to do with the lateral surface. What the lateral surface of the cells is by gap junctions. Gap junctions. Okay. And the next one is the examples of locations. Okay, where do you find the examples of locations? Where do you find these cells in the tissue, in the body? It present in the kidney tubules, okay, and the closed body cavities, okay, and mesothelium, that is the derivatives, and the lining of vessels, we call it as endothelium. The closed body cavities, this is derived from mesothelium. Okay, mesothelium is the one which we discussed earlier with the picture which I showed you. I'll recollect you what is mesothelium here. You see that one is a mesothelium, lines of closed cavities. I will come back a bit later on that. Derived from mesothelium, and also this simple squamous epithelium is present in lining of vessels. Lining, lining of vessels. Blood vessels is the lining, okay, and that is called again is an endothelium. So endothelium is present in the blood vessels. Like this is the blood vessels is going. This lining inside this lining, and this lining, the cells are present in this lining. We call it as endothelial cell. I'll give you that one. You can see that one here. The cells which are present here. These are blood vessels. Blood vessel. Capillaries are blood vessels. These cells plays a major role in this endothelium. If anything is happening to these cells, any injury, it can anchor a lot of cellular injury on the blood vessels, and more of extra cholesterol, and it is not metabolized, and the excess it is going to deposit here. That for the atherosclerosis and other complications. So the endothelium is the one which is more of vascular regeneration, meaning blood vessels growth, vascular regeneration. Okay, vascular regeneration. This all because the examples of location. What we are looking now is the simple squamous epithelium. Simple squamous epithelium are present over here. Okay, what is the functions of this simple squamous epithelium? What is the functions? Coming back to the functions. Okay, good. What is the functions of this epithelium? Main functions are absorption, of water, okay, protection, diffusion, okay, of what? Diffusion of oxygen and carbon dioxide, simply a gas exchange, and protection and absorption, okay, absorption of water. So these are the main functions of simple squamous epithelium. And where this tissue is derived from the embryonic origin. I'll put it over there, like an embryonic origin for this simple squamous epithelium. Simple squamous epithelium. The embryonic origin is from mesoderm. Okay. I'll explain this in quite a bit now. When the fertilized egg become 23 plus 23 chromosomes to form a fertilized egg at the time of conception, after the it is going to divide into 2 cells, and then 2 into 4 cells in the embryonic development, and then 4, and then you got an 8 cells, and it's going on, and then finally it formed the first type of the layer, which is coming out, if the development of embryo, I just write is the ectoderm, and then the middle one is the mesoderm, and then you have another one is the endoderm layer. So this is one layer of cells, this is another layer of cells, this is another layer of cells. I put a different color. Like this one is the external body surface, like ectoderm. So these are the cells which are present outside of the body, and the derivatives of that, ectodermal derivatives, and some of them are the mesoderm, that's on the middle with the muscles and other cells, like this one is from mesoderm. Another one is the endoderm, okay, it's inside. So these are the 3 layer, otherwise a tri-layer. So always you have to classify whenever the tissue is there, whether that is the origin from either from the ectodermal origin or the mesodermal origin or endodermal origin. Okay, and these cells, simple squamous epithelium, is coming from the mesoderm origin of the embryonic development. So that's another thing.
Which is the middle one, right?
Yes, that's right, the middle one. Correct. Right. The comments for these particular cells, what we have a comments, mesothelium, what the mesothelium, okay, facilitates movements of viscera. Facilitates movements of viscera, that's the abdomen, okay, viscera, and may regenerate from adjacent connective tissue cells. They also, regeneration capacity. Regeneration from adjacent, ADJACENT, adjacent connective tissue cells. We will see later on connective tissue cells. So all we are talking about the comments. What is the comments for? These comments for the simple squamous epithelium. Again, we are dealing with the squamous epithelium. So up to this, we have covered all the topics like morphological type, shape of cells, number of layers, specialization of free surface, lateral surface, basal surface, examples, and function, and embryonic origin, and comments. We have to classify all the tissues on this category. Okay, so we'll see one by one again. The next one, what we need to do is the next type of cells in the epithelial tissue. Next type of cells is the stratified squamous epithelium. Okay, stratified squamous epithelium. Type is stratified squamous epithelium. squamous epithelium. Okay, stratified squamous epithelium, and this is non-keratinized. This we will classify this, and then we'll go non-keratin, Keratinized, non-keratinized. Now the keratin is not present in these cells. Stratified squamous epithelium, and the surface cells, the shape of the cells. The shape of the cells for the stratified, it's a flat, deeper cells. I'll write it like a. This is for the stratified cells. The type of cells is a flat, but it's a polyhedral. What is polyhedral? Or polyhedral? Tetrahedron, polyhedral. Like the cell shape, like this. It goes like this. You have a different shape of those things. Non-keratinized cells. The cells like this. It looks like this. This nucleus. You see that one here? I mean, these cells have been attached with each other with the with the junctions and everything. We will see that one. A number of layers. The previous one, the simple squamous, we have only one layer. But the polyhedral flat and this particular stratified, you have many layers, 2 layers, or many layers, many. Okay, I'll write it here, 2 layers or many layers. That is for the stratified squamous epithelium, the shape of the cells. Okay. Now, go on to the next characteristic of the cells is the number of layers. That's why we said 2 or many layers. 2 or many layers. So this is going along with this. 2 flat polyhedral, but 2 layers or more layers. Okay. The next topic of the characterization, specialization of the free surface. This free surface is again microplasia. It is modified as a microplasia, and the basal one, which is the basal surface modification, especially in basal surface. That's which I discussed about the earlier. The basal surface is the basal one. This is again hemidesmosomes. Write it as hemidesmosomes. So they are nothing but the cell junctions. Cell junctions. Junctions. The proteins. There's a basal surface been attached to it. And the next one, lateral surface modifications. Okay, the lateral surface. What is the lateral surface for this particular stratified squamous? You see desmosomes, and they have the cell layers between the basal and the intermediate cell layers, or the lateral one. This is the lateral one. This is again, this nucleus, and this has been attached with the modification with the desmosomes. Desmosomes. Okay, desmosomes. And where do you find these cells? That's examples of location of these cells. They are present in, I'll write it another paper, lines of oral cavity. Lines of oral cavity. Lines of oral cavity, esophagus, 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 okay, and vagina and anus. So these tissues, I mean, the example of location, what type of tissue is this? It is stratified squamous epithelium. Okay, tissue is stratified. That's what we are looking into. Stratified, all these characteristics of stratified squamous epithelium. Okay, the next one is the function. What is the function of the stratified epithelium? Functions goes along with protection. Protection from any other infections and other, because it's present on the external surface and it secretes and a lot of layers, so it can protect our inner parts of the tissues. And the next one is the embryonic origin for this particular stratified squamous epithelium. Embryonic origin is endoderm and ectoderm. Endo and ectoderm. This is from embryonic origin of this layer. Okay. And especially the comments for this, on this particular stratified squamous epithelium, people are often confused with because of the multiple layer, due to multiple layer, due to this multiple layer of the cells, this is often confused with the transitional epithelium. We will see a bit later. Transitional epithelium. Okay, often confused with the transitional epithelium. This is the comment for stratified squamous epithelium. Now we will go on to the next one, which is a stratified keratinized one. But most of those, the findings what we have seen, the previous one is the same one, but except some keratinized cells are present in that. Okay, we'll go on to. I'm not going to display all of them for this, but please remember that another type of stratified keratinized, this is a keratin, this is a protein keratinized, stratified keratinized squamous epithelium. Just like what we have seen the previous one, the same type, but except there is the free surface of is keratinized, okay, and the superficial layer is rich in keratin, KERATIN, keratin. All of them are the same. And the location where you find this stratified keratinized epithelium, the epidermis. Okay, present in epidermis layer, right here, epidermis layer. And prevents dehydration, protection, and synthesis of vitamin D. If you take a sun bath or going to the beach and getting excess sun, and this epidermis layer, that is the stratified keratinized squamous epithelium, produces vitamin D. That's the major functions. And it prevents the dehydration. When you it secretes and keep the cells or skin moisture. And main origin This cells is embryonic origin. This is coming from ectoderm. Ectoderm is the outer layer, and the comments for this is the effective barrier. It acts like a barrier. If you look into your skin, that will give an idea, like external barrier for any toxic chemicals or substance. Protects toxic substance from environment. So if you work in petroleum industry or somewhere, more chemical industries, you are likely to get exposed to different solvent systems. And if you have an injury to the surface of the cells or the skin, this substance can easily penetrate, getting in. So you should be very careful to avoid that. Also, we explain in our lab to wear protective clothing and gloves and everything, right? Okay. Now what will go, the next one, the type of epithelium we are going to discuss is cuboidal epithelium. Okay. We are going to discuss much on cuboidal. Cuboidal epithelium. Okay. And this cuboidal epithelium, it's classified into two parts. Number one, cuboidal, simple. It's a single layer. Simple means you have a single layer of cells. Single layer, just monolayer on that. The another one is stratified. Stratified is a multiple layer, multiple layer of cell. Like here, it goes like this. Cells in a cuboidal cells, and cuboidal cells, as I mentioned in the lab as well, the structure of the cuboidal cells like a cubic, right? These cells, and then another cells will go like this, and another cells will go like this. So if you see the cuboidal, if it is one layer, okay, that's on the simple cell, and if it is a multiple layer, another layer, another layer, another layer like this, and another layer. So if it goes on that, that's called stratified multiple layer of the cells, the stratified tissue. This is a simple cuboidal tissue, stratified cuboidal tissue, and this one usually 2 to 3 layers, multiple layers, 2 to 3 layers of the cells. Okay, and the cuboidal cells and specialization on the free surface. What is the free surface of this one? This is the free surface of these cells, cuboidal cells, and they have, with the microvilli, they have the free surface. This is, we are talking about the cuboidal cell, nucleus, and then this surface of the cells. This is cuboidal cell. This surface with a microvilli, just going over there. This is a modified one. Microvilli. Okay, that's on free. And this is a fuzzy coat, like a fuzzy coat. Present over there. And the extensive basal foldings, suppose in the basal surface, this is the basal surface which I explained earlier. This will have a folding like this. This one, basal surface, external folding in convoluted tubules, and this is the same cells, but this membrane or the basal membrane, it goes modified like this. It is not like, you just ignore this one, but the same cells like this, it goes like this. So where do you find it? So one side is a microvilli, the another side is, this is something like the lumen. It present in extensively as a tubules of kidney. You find it in kidney. You find it in the kidney. Okay. So these are the basal cell and this is a free cell. Now what happened to the the lateral surface of this one. Okay, you have some desmosomes are there here, and because these are desmosomes, is the tight junction protein, and this is the lateral surface desmosomes. Basal surface, we convoluted. Is a convoluted. C-O-N-V-O-L-U-T-E-D. Convoluted. Because this is going the folding and folding. The lower membrane, cell membrane of these particular cells is being folded, and it helps the kidneys and transport across. One side is the absorption, the another side it will reabsorptions and everything is happening here. So in the kidney convoluted tubules, C-O-N-V-O-L-U-T-E-D, we will study on the function of these cells when you do on comparative physiology class or something like. At the time physiology, you will see the functions and what type of proteins are there. But we will see briefly later on this one too. The function of this, secretion as well as the protection. Okay. Before that, where do you find these cells? Where do you find this type of cells? You will find in thyroid follicles. Thyroid. They are thyroxine hormone synthesized, so thyroid follicles, and the kidney tubules. Okay, you will find, and the function for this tissue is secretion and protection too. Secretion and protection are the functions here. And where you get these cells, origin, endodermal as well as mesoderm, embryonic origin, where this simple cuboidal epithelium is coming from endoderm and mesoderm. Okay, and this tubules are the kidney tubules and the other tissues are simple cuboidal epithelium is common in kidney. It's prevalent in kidney. What functions in the kidney? What the kidney will do? Kidney mainly on the filtration of the waste products like urea, and if it is excess in glucose or nitrogenous and phosphate and calcium. Most of the nitrogenous waste products has been removed by the kidney. It's a filtering mechanism. So these cells are adapted in such a way it does the convoluted tubules, it absorbs and then excrete the compound. So the main function of this simple cuboidal, it's a secretion as well as the excretion product, okay, in the kidney. Also, it is present in salivary gland. So if you collect some saliva or the oral swab, you will get this cuboidal epithelium. We are going to do it in our lab. I'll ask you, everyone in the lab, to take your oral swab and stain it and see under the microscope, and then we will observe it. Okay, what type of tissue? Because some of those tissues or cells are coming into saliva. So salivary gland that's rich in cuboidal epithelium, simple cuboidal epithelium. Also, you get in pancreas, where the insulin is synthesized. Okay, that's in pancreas. Also, you get simple cuboidal epithelium. Okay, the next one is the cuboidal epithelium, but multiple layer of. This is a single layer, but we will see the another one as a multiple layer of cuboidal. Another type, as I mentioned before, cuboidal epithelium, but this is a stratified cuboidal. No, yes, I'll write it again. Stratified. S-T-R-A-T-I-F-I-E-D. Stratified cuboidal epithelium. And the cells, like a cuboidal, and usually how many layers? As I mentioned, 2 to 3 layers. And the specialization free surface is not much. There is no specialization of the cells, like there is nothing there, you know, and as well as in the bottom is like a flat. It's unlike the simple one where the convoluted tubules, there is no convoluted or any other modifications of the basal region. And if you have the lateral surface is the desmosomes, is the same one what we observed the other one. And the next one, where do you find this one, the cuboidal stratified or multi-layered epithelium? Where do you find? You will find in sweat gland. It's W-A-T, sweat gland. Sweat gland ducts, you know, the passage. Sweat gland ducts. And the developing ovarian follicle, ovarian follicles. They are made up of multiple layer of the stratified, multiple layer cuboidal epithelium. And the main function is, again, the function is the protection. Protection is the function, and also secretion. And the origin is the same thing like a mesoderm, but also it also derived from ectoderm. So embryonic origin is a bit different. One is the same, but the other one is a bit different when compared to the simple cuboidal epithelium. So the embryonic origin of this tissue is ectoderm and the mesoderm. So the simple cuboidic, you have endoderm and mesoderm, but the stratified cuboidal epithelium, you get ectoderm and mesoderm origin. That you should remember that. And it's you know, these are bit rare types, but you know, it's not very when compared to the other one, that the previous one. So where you have the kidney and salivary glands and pancreas present for the simple, but you won't get prevalent for any other region except what we discussed earlier, sweat gland, ducts, and ovarian follicles. That's this is a simple cuboidal. The next one, of the type of epithelial tissue, we have to discuss about the columnar epithelium. Okay, columnar, C-O-L-U-M-N-A-R, columnar epithelium. So we have covered now. There are two types of epithelium. That the first one, what we cover, is the squamous epithelium. Second one is the cuboidal epithelium, and different types. Now we are going to discuss about the columnar epithelium, and there are 4 types of columnar epithelium. Okay, we will discuss one by one. What are the 4 types of the columnar epithelium? Number 1, simple. Again, what is simple means? Only one layer. And then second, we are going to discuss about the stratified or multiple layer, stratified epithelium, or layer after layer. And then 3 is pseudo-stratified. E-S-E-U-D-O, pseudo-stratified. Pseudo-stratified means it looks like a multiple layer, but it is not. Okay? And the another one we have to see is transitional. Transitional epithelium. So, the simple columnar epithelium, stratified columnar epithelium, pseudo-stratified columnar epithelium, transitional columnar epithelium. So, all of them will go the next, like a middle name and last name. Last name is epithelium, all the 4. Middle name is here the columnar. First name is may vary, like a simple, stratified, pseudo-stratified, and transitional. So you keep remember like a simple columnar epithelium, stratified columnar epithelium, pseudo-stratified columnar epithelium, and transitional columnar epithelium. You will see one by one and what is their characteristics. Okay. Okay. What is the first one is simple simple columnar epithelium. Simple columnar epithelium. I short SCE. Where the shape of the cells for the simple is the various heights. The columnar means like a column. See, like this. Okay, and this is the nucleus, right? Then another cells looks like this, going. Another cells like this. It's a big column of that. You know what do you mean by column? Like, isn't it? So now we are going to see if it is present in a one layer, that is the simple one. This one layer as a monolayer nucleus. Now you have the basal surface here, basal surface, lateral surface, and this is the free surface. So we will see the modifications now. Okay. Simple, you have only one layer, one or monolayer. Just only one layer of the cells. Okay. The free surface, which is being modified as a you have a stratified border or a prominent or a fuzzy coat or you have got this is modified like this, just I'm just writing this as a microvilli. See, columnar epithelium, you have a microvilli on the free surface. Microvilli. Okay. And the basal surface here, it is being modified as hemidesmosomes. Hemidesmosomes. I'll write it here if you can't clearly see that. Hemidesmosomes. This is a basal, a type of protein, a cell junction protein, which is being used to attach the basal other tissues. So that's why the base is modified in the hemidesmosomes at the bottom, the basal surface. And the next one, if it is present in the lateral surface, is the desmosomes in the lateral surface. Here, in the lateral surface, is desmosomes. DESMOSOMES, desmosomes. And they also call it as zonula occludens. I'll write it in a separate paper. We got like a zonula occludens, OCCLUDANS. These are some specialized proteins. I'll just draw like this. Okay, this is the cell, and these are another cell. Okay, these are another cell, and these cells and these cells been communicating with each other with these zonula occludens, been attached. Okay, so occluding, joining, and zone. The site where it joins together. These are zonula occludens. They got different types of protein. We got zonula occludens 1, like a ZO1, ZO2, ZO3, like that. So, for example, in a cholera toxin, will abolish some of these zonula 1, ZO1 or ZO3. So what will happen? The cholera toxin abolish this junction and make the tight junction become a loose junction, and thereby it allow all the water which is present in the bloodstream and passing into the lumen of the intestine, and you will get a watery diarrhea if you have a cholera. So cholera toxins specifically inhibit the junctional complex or zonula occludens. So that's very important to know about it. Okay. Now, as I mentioned before, where you find these cells, these columnar epithelium with the microvilli, the absorption surface area, everything, you will find in, so just now I explained to you, lines of gastrointestinal tract, GI tract. Okay, and the functions, this is the location you find the gastrointestinal tract. The function is mainly of secretion of enzymes, secretion of proteins, and sometimes some hormones. Here also it gives a protection, GI tracts in protection, right? Also in absorption, the function is mainly of absorption. Function is absorption and lubrications. It gives some lucus or mucus being secreted with the lubricant. Lubrication. So my earlier research, I extensively work on the absorption of nutrients in GI tract. Okay, So that's my specialities, and then I did extensive work, and I will show a couple of those slides to you later on. Okay, so the absorptions mainly with the what type of absorption they will do, like a glucose and amino acids and fat or fatty acids, and then you have vitamins and minerals. So I did work on glucose absorption, amino acid transport, and fatty acid, as well as the bile acid transport. So all this transport, I did my own research for to probably more than 10 years of my research on this, all these transport mechanisms on the epithelial cells. So it's quite interesting, but we will discuss a bit later on. But just want to mention you about the function of the columnar epithelium. Okay. Now the embryonic origin for this particular columnar epithelium is from endoderm layer. That's an embryonic origin. And also there's some comments over that what is the function. In addition to that, they also present in goblet cells, G-O-B-L-E-T, goblet cells, and they secrete mucus. They are present, as I mentioned before, where I have got it. Okay. So these are the columnar epithelium. These cells are present in between these cells, like this, like this. Okay. These cells are goblet cells, the green color, and they secrete what? Mucus. That will do the lubrication function in the GA tract, the part of the things. Okay, that's the part.
But
Now,
are they also columnar?
yes.
The goblet?
No, goblet cell is not a columnar cell. No, it's a independent cell, but it is present. A small cell. It is not like a column like this, but it's a small cell, but present in between the junctions. Okay. Now let's go on to the next type of columnar epithelium. That is on the stratified columnar epithelium. Okay, we will see the second type, like a stratified. I mentioned earlier, the stratified is the multiple layer columnar epithelium cells, multiple layer of the columnar epithelium. And they normally used to present as a 2 to 6, and they have a columnar but deeper cells. Suppose, as I mentioned, this is the multiple layer. See, this is the one layer, okay, and this is another layer. And what happened when this layer, which is going down deep, become cuboidal, but still it is this nucleus. I'll just put a dot on the nucleus. Could you see that this is a length here, and this length becomes shortened a little bit, and then it is shortened, and finally it looks like cuboidal. Looks like cuboidal. But it is not. It is a columnar epithelial, but you can say a short form of columnar epithelium, short column, and this is a long column. So it is a multiple layer. 3 layer or 6 layer. So when you go down, you looks like a cuboidal cell. Yeah, you also present where the free surface of this is a small microvilli. It's not a long one as a GA tract, but they are present in, you can call it a short microvilli, not long one, microvilli, on the free surface. Here, they are. This is the basal one where the columnar. So because it's a multiple layer, okay, but this a short microvilli are present here. Where do you find in the short microvilli are present urethras. U-R-E-T-H-R-A-S, urethra. Urethra, you find the short microvilli of this one, these cells, okay, and the specific basal surface is the same as we discussed are the hemidesmosomes here. This has been connected, okay, and the lateral surface is same junctional complex like a zonal occludens, which I mentioned earlier, and the locations where you find on the location here. Here location is the urethras, male and female urethras, and also it's present in mammary gland. In mammary gland where you find the stratified epithelium. And the embryonic origin for the stratified, it is mainly from, I write it here, endoderm. Endoderm. Embryonic origin from endoderm. Okay. Now we will go on to the next type of columnar epithelium. Is pseudostratified, otherwise called ciliated one. Okay, that's the third type in our list as a pseudostratified.
Is there a function for the stratified columnar?
The stratified, which I mentioned earlier, and it function as a just a second. Protection. Okay, function as a protection for the stratified epithelium. So now we'll go on to the third, that's on the pseudo, Pseudo, stratified, stratified, pseudo-stratified epithelium, but here we will study specific to ciliated one. The cilia are present. And here the cells are varying in height but all rest in basal lamina. You just watch here. This is the basal lamina. Okay. One cell looks like this because these are columnar epithelial cell. This is one cell. Okay. The another cells looks like this. Another cells will go like this here. Another cells like this, a short column. The another one is a long column. Okay, it goes like so. If you see the whole cells of the tissue in a microscope slide, you will find a multiple layer, but actually it is not a multiple layer. All these cells are attached to the basal lamina from this origin, but they clumped and the shape is not like a straight one, but they go on just a bend and then one cell over the other, but it looks like a stratified or multiple layer, but actually it is only one layer. Okay, that's why we call it as a pseudo stratified epithelial cells. And then this is the columnar epithelium. Also it is the cilia which is present at this one. Cilia mainly present in tracheal tubules. That's all the found where we found, and we also discussed in our lab as well, right? And these are the cilia. This is the free surface again. It's the cilia in the free surface. It's modified, and sometimes the microvilli is interposed, but the basal surface is the basal lamina with hemidesmosomes here. Hemidesmosomes. I'll write it here. Hemidesmosome. Desmosomes. Same thing. I'm writing again and again, so please don't worry about it. And then the lateral part is again the zonal occludens here, lateral surface. Where do you find this one, ciliary pseudostratified? You find in respiratory passage. What is called? Trachea. Okay, respiratory passage. And the function here again, protection, and elimination of, simply I'll write the function here. Function like an elimination of airborne dust. Okay, airborne dust. B-O-R-N-E, airborne dust. I write it here. Airborne dust. Okay, it activates while and also secrete mucus. M-U-C-U-S, secrete mucus, and which also help move to the mouth by ciliary action. That's why if you have a nasal secretion and if you have a strong infection, what will happen? The nasal secretion getting into your throat. It's pushing back. So the ciliary action, violent action, will using the mucus and the airborne dust and everything coming to the mouth, and then you have to spit it out. And the next one is the embryonic origin is from endoderm. Endoderm origin. Okay. And they also call it as a respiratory epithelium. So in the other words, pseudo-stratified epithelium, ciliated epithelium, also called respiratory epithelium. Respiratory epithelium. Respiratory epithelium. Okay. Now, the last one is we are going to see as a transitional epithelium. Have a transitional. Okay. Number 4. Transitional epithelium, and these they have the what type of cells there, and the surface cells are dome shaped, as in present in the empty bladder, flat in the extended organs, and basal cells are polyhedral. So they are stratified. Okay, stratified. Maybe you have a multiple layer. Okay. And the surface cells are dome-shaped, and then the bladder, urinary bladder cells, where you find the stratified epithelium, gives more of the elasticity. It's not due to the collagen, but because of the multiple layer are crowded, and then it can elapse. I can write it like this. These are the cells, something like this. It goes on more crowded and crowded. And multiple layer, okay. Yeah, I'll do that. Okay. These cells, okay, these are the columnar epithelial cells, but it's crowded. But if you suppose if this is going on a urinary bladder, or the cells, if the urine is accumulating here, these cells will expand, and then it will stretch to this end, up to this. Initially it was like this, but it can extend. At that time, the size will also a bit larger. So it looks like elasticity, but it is not elasticity, but the tissue it's a very thick one, or it is crowded, but when it swells, but because accumulation of urine in the urinary bladder, it can expand. So this type of the urinary or the bladder modified stratified epithelium, otherwise we call it transitional, because it can, when the urine is wide, and it will come back to the original. So it contract and then relax at these in urinary bladder, contraction and then relaxation. So these cells mainly are transitional type. Okay, and they are present in the lines of urinary passage. Yes, and they also have the same type of free surface as well as the basal surface. And the epithelial origin for this transepithelial origin is from mesoderm. Mesoderm and endoderm. Okay. And the adaptation, the main function here, the adaptation for the distension cells, right? Adaptation. And for the distension, the cell size will increase and decrease. Distension of cells. Distension, T-I-O-N, distension. Okay. By cells are sliding on over the other. Cells are sliding. This epithelium on the layer. So that's why it contract and relax. So what I'll give this one here on mainly onto the epithelial tissue right now. So, if you have any doubt from any centers, please, from Sugarland or Victoria, any questions?
For this one, would the function be elasticity
So the next lecture is about the connective tissue. Okay. Connective tissue. So the major tissues as the primary tissue which we discussed earlier, epithelial tissue, connective tissue, muscle tissue, and nerve tissues, right? So that's what we did. Right. I'll show you here again. Okay, now here you can see the first epithelial tissue we finished now, and now we are going to our connective tissue, and then we will go on to muscular tissue and nervous tissue later on another week. But I'm going to finish it up the connective tissue today. Okay, the primary tissues. So the connective tissue characteristic, what this connective tissue will do? The characteristic of connective tissue provides binding to what? Binding, okay, anchoring. Anchoring, okay. Binding and anchoring means been attaching, okay. And filler, and also it fill. Otherwise, it's the filler cells. Filler. For what? For supporting tissue. For supporting structure of our body. So the connective tissue plays a major role in attaching a type of one tissue into the another tissue. So one organ to the another organ. In our body, you get different types of cavities. You know, if you have one particular organ to the another organ, what will happen in between that? You know, you need to fill something like. If you make a parcel for a Christmas gift in a box, what you do? You keep it in something like a glass, and it is safely reach the destination. What you need to do? What you do for your safety? If you keep the things with what? If you want to transport a wine glasses to make that you are giving a gift to somebody else with a wine bottle. So you are keeping a bottle over here, right? Wine bottle for Christmas. And you have got the wine glasses as well. And if you want to buy, and if you want to send it out. Yeah, you stuck with what? Beanies. And you have got some papers, and then you have a filling, right? Make a cushion like this, right? Even the US PA or the Federal Express and these guys are throw this one. They don't care because there are thousands and thousands of packets, right? The packets too, they throw them out. So at the time you find, even if you say the glass with the care and then up, this is a down and all marked, and they don't care. You know, you are caring because you should reach the destination properly without any breakage. So this is, you are taking the same thing in our body system. If you have this, your body, suppose if I draw a sticky man, like, you know. So if you go and see inside, inside the body part, you see you have, bones and tissues and also, different organs like heart and lungs and intestine. So they are arrangement. The arrangement is is quite fascinating. So all of them are being connected, to keep it in a safer situation. That's mainly on to the filler and supporting structure. That is the purpose of this connective tissue. Okay, what type of cells which they do, and where the origin or the embryonic origin of this connective tissue? The origin is again is coming from is mesenchymal cells. Mesenchymal origin, embryonic origin. Okay, origin, origin, origin. Some of them are derived from glia cell, glia, G-L-I-A, glia cells. Some of them, some connective tissues from here, but mostly it's coming from mesenchymal cells. And what is the constituents of the connective tissue? What the connective tissue constitute, or what constitute the connective tissue? Constituents, S-T-I-T-U-N-T-S, constituents of connective tissue. They are, constituents are, I'll write it here. mostly fibroblast, mostly fibroblasts. Okay, and number two, they made up of number two, extracellular fibers, extracellular fibers. Okay, and these extracellular fibers, what type of fibers we have in our body? The reticular fiber, reticular, reticular fibers. And another type of fiber is collagen fiber, collagen. And another fiber is elastic, all fibers. Okay. And another type is the ground substance, See, number one, fibroblast, two, extracellular fibers, and ground substance. What are the ground substance? Here is the tissue fluid, ground substance. Tissue fluid is a ground substance. Gel-like material, gel-like material, and solid as cartilage bone, solid, like cartilage and bone. So the ground substance, like a solid ground substance present in cartilage, and ground substance like in bone, that's also solid. And somewhere you get a gel-like structure, that's also ground substance. And then the tissue fluid, whatever the fluid we have it, even including blood, right? They are the ground substance, tissue fluid and interstitial fluids, they are the ground substance. So the connective tissues, it will connect. Suppose here the tissue fluid connect from one organ to the another organ. It will move from one place to another place. And also it connect the tissues of various, suppose if the tissues, intestinal tissues absorb the nutrients and also the blood contain oxygen. All of them are using the tissue fluid as a connecting fluid. It connects different organs to the brain and to the arm, to the muscles, and to the distant part of our body. So the fluid plays a major role. The same thing is if you have a cartilage and bone like a solid, if you have the ground substance like a, without the bone become like a gel-like thing, the ground substance become in the bone like a fluid, what will happen? You will become a gooey, like a jelly, jellyfish, not even jellyfish, but you will become like a solution, right? So you won't be as a personality, a structure. So we have to have this ground substance, solid. So we will see all on the how it been connected. Okay. Let's go on to the next one. How do we classify this? Classify the connective tissue. Classify. We can classify these connective tissue as amount and type of matrix. Depending upon amount and type of matrix. Okay. And then we can also classify the connective tissue depending upon the type and arrangement of fibers. Arrangement of fibers. We can classify. And then the abundance of kinds of cells, the type of cells, some kind of cells more, some of them are less, so abundance, A-B-U-N-D-A-N-C, abundance of cells, the connective tissue cells. So we can classify depending upon these criteria. Okay. And some of them are classification which is going on transitional connective tissue that forms, they're based on the body needs. Sometimes, the body, depending upon the requirement, the need of the body, the connective tissues, it may vary. Like I can write like a transitional connective tissue. Transitional connective tissue, which will go like based on body need. Based upon body needs, these connective tissues, they classify as a transitional connective tissue. Meaning, if there is a need, that particular connective tissue will be present. Otherwise, it will not present normally that particular tissue. They form like a, again, loose connective tissue or dense connective tissue. Loose or dense. They have regular connective tissue, regular or irregular. I like this pair, easy to remember. Loose or dense or regular or irregular. Another pair is elastic or collagenous. Elastic or collagenous. C-O-L-L-A-G-E-N. Collagen. Elastic or collagenous. Depending upon the fibers. Okay. These are the transitional. Now we will go on to the classifications on the loose connective tissue, otherwise we call it as a areolar connective tissue. Another name, loose connective tissue or areolar, A-R-E-O-L-A-R, areolar. Meaning, you get more of a gap in between that, just like we fillers, these connective tissues, as more delicate, and they also present in the beneath the mucous membrane. These are the loose connective tissues. Okay, that's a filler between the organs. I'll explain in a different sheet. We have loose connective tissue. Something like in the package, what you have, this is your gift, okay? Your gift. So you don't want to break, so something like, so what you normally do, you have to filler, right? The filler should go like this. You should have a spongy, like a sponge. Like the red is a sponge like that, and it's the filler. Okay, like this in our system, the loose connective tissue will act like a filler between the organ. This is the one type of organ. This is another organ, and these are the red one is our areolar or the connective tissues. Okay, and it classified as a delicate network. The loose connective tissue formed as a delicate network in our system. Delicate network. And they are present beneath the mucous membrane. Okay, present in beneath mucous membrane. M-U-C-U-S, mucous membrane. Beneath the mucous membrane. And they also form like collagenous fibers and elastic fibers and cells of the fibroblast and macrophage. These loose connective tissue, they have a different types here. Okay, again I'm going down to the loose connective tissues. Loose connective tissues or areolar tissue, they form the network. How the network will be formed? You need to have a fiber, right? If you have a net means you have got different fibers, and then another type of fibers will going like this. Okay, then in between that, what you get, some of the cells over there, cells. These are the cells or the connective tissue cells. Okay. So some of the cells are here. So these are fibers. I'll give you the fibers. Different types of fibers. Here we know about the two types of fibers. We know about the elastic fiber or collagen fiber. Collagen fibers. Okay. And then we also have cells. What type of cells we can say now? The type of cells is macrophage. The cells. I'll write it in another sheet of paper. The cells. Cells consist of the fibroblast cells and macrophage. And also, you have a mesenchymal cell. Mesenchymal cells. Okay. And the fibroblast will produce more of collagen fibers. Fibroblast, collagen fibers. The macrophage is there, and this macrophage is derived from the mesenchymal cells. It will produce macrophage. Mesenchymal cells produce macrophage, and the mesenchymal produce osteoblast for the bone, osteoblast, and mesenchymal cells again produce chondroblast. So, when you see, I'll come back later, this one, the loose connective tissue is made up of fibers, and these fibers is derived from however these fibers are synthesizing is coming from fibroblast cells. These cells, one of the cells will produce fibers, and one of the cells produce elastic fibers, and and then these fibers is coming again from the collagen. The collagen is coming from again from the fibroblast. So the cells produce this protein as an extracellular. It produces the protein and export outside, and then you form this network, and thereby you get a connection. Connective tissue perform its function. So some of these cells, osteoblast, a bone cell. Some of the cells present in the connective tissue as a chondroblast, where you get the the cartilage. So the bone and cartilage. All of them derived from the mesenchymal cells. Some of them are the macrophage. So predominantly the fibroblast for the fibers and the mesenchymal cells for the other substance can be secreted out. So these are present as in a fashion as a loose connective tissue. Example of loose connective tissue. Okay. The next one, we need the types of cells again in the loose connective tissues. We have done two cells. Okay, we have other cells too. Types of cells. Resident cells, which is resident cells. What is resident cells? Resident cells which will produce, it's a permanent residence on the connective tissue. It will produce the fibers, like a fibroblast cells as a resident cell. Then macrophage as a resident cell, resident cell. And then mesenchymal cells, which we'll discuss earlier. And then reticular cell with the reticular fiber, reticulum, R-E-T-I-C-U-L-A-R, reticular cells. These are cells. All these four cells are permanent residents in the cells in the connective tissue. The another type of cells in the loose connective tissue is visitors or visitants. They will come only there is a need. Visitant cells. I'll write it in a separate paper. Number 2, visitants. Visitant cells. Visitant cells are the plasma cells. Plasma cells. Another one is the mast cells. And leukocyte, L-E-U-K-O-C-Y-T-E-S, leukocytes. And pigment cells. Okay. Now we will discuss about this type of cell. Then we will have a comprehensive knowledge of the entire connective tissue chapter. Okay. Types of cells here for the loose connective tissue. Resident cell will produce all the fibers, and macrophage will fight against any infections or anything which is coming inside our system. The macrophage may vary organ to organ. For example, in liver it is called the Kupffer cells. Okay, in brain it's called Schwann cells. So and then the mesenchymal cells will give rise to the macrophage, as well as other substance like osteoblast and chondroblast, for the bone and ground substance it will produce. So it is a permanent resident on the cell. It is necessary, a type of cell. And then reticular fiber, which will produce the reticular cells. So these are the resident cells. That some of them are visitants. When you get an infection, suppose when you have a series of some antigenic, or if you have some vaccination, or if you have to protect in your body, then the visitant cells will come into play by from the blood into the connective tissue, and the plasma cells is the one which will produce more of antibody. If you are taking immunology lecture, immunology class, you know about the role of the plasma cells. Also, mast cells will come if there is any injury onto the tissue, and nearby the mast cells will eliminate as a histamine release to sign, to give a 911 call to the body to give more blood flow to that particular region or the site of inflammation. The histamine will be released from mast cells. So mast cells will migrate towards the point of injury, and so that's the visitant cells. Also leukocytes, because of mast cells, and it will eliminate the histamine, and the histamine will do vasodilation, and vasodilation will attract more of white blood cells or leukocytes. Leukocytes, the white blood cells, will reach the site of injury. And then another type of cells is the specialized cells, where the pigment cells. The pigment cells will migrate. This is something like the tanning of suntan. Sometimes if you go on tour, so you are activating some of the cells below the skin, and that the pigment cells will also produce melanin, and that will come and to play a major role. These are the visitant cells. That's why when you have a suntan during the summer, and then after that what happened? Your suntan coming to the original. That's a transient. That's a visitant. It will go away unless otherwise you have to have a constant suntan or tanning club or spa or something like that. You have to go on permanent. Okay, coming to that point. So you have loose connective tissue. We have discussed about the two types of resident cell and visitant cells. Okay. Now let's go on to the adipose cells. Another one is the interesting one, like an areolar cell, is the adipose or fat cells. Adipose tissue. I think we have discussed extensively in our lab, but the adipose tissue is something like a big vacuole-like thing, but it is not a vacuole. But this is your membrane, and you also have the green color, which is going. I'm doing this one. Is the cytoplasm, and you have a nucleus over here. The red is the nucleus. Nucleus is the one side pushed, and the middle part is the lipid storage. Okay, and this is your adipose tissue, lipid storage, right? So the functions and everything we discussed about it, because it's the storage mainly, and also it gives an idea about the insulations and everything on this part of the body, right? So this is a type of cells that is also derived from mesenchymal cells. Okay. Okay. Plasma cells, yes, we know about this. So see, I think I have given these pictures into your figures, into your chapters. You can see this ectodermal layer of the melanocytes when the cells will transient. If the synthesis of melanin, it will go out. So this is something like activated by UV, and then more of melanin synthesis. That's what is going on the tan. Okay. So these are the melanocytes synthesize the tyrosinase and oxidase and tyrosine dopa, and dopaquine, and melanin. So these are the activation, and these are transient cells and melanin synthesizing cells. That's also visitants, which we discussed earlier. Okay. And then the support function of the extracellular fibers, which we discussed earlier. What is the role of the fibers in this case? Fibers. Yes, extracellular fibers, collagen, reticuloendothelial cells, isn't it? So the function mainly of the fibrous capsules of organs. Suppose I want to house an organ here. Okay, this is the dark one, like this. Okay. So I want to house, this is a type of organ which just I want to, make a house, keep it safe, right? I want to make a cushion of it. So what happened, these cells, which is writing here, these cells make a house for this particular organ, okay? So it has the fibers, it here fibers, And they penetrate the tubercula, penetrating the organ. It gives some sort of a canal so that it gives the connections and keep it cushion on that. And also you get this type of fibers it present in skin. Okay, and ligaments and tendons. Okay, ligaments and tendons. Again, and then we also find in the bone and cartilage. See, bone, cartilage, and ligaments and tendons, they all do the function as a protection. Bone will protect the inside organ like a heart and lungs, give the protection. And the cartilage, which is the present in between the bones, it should not get cracked out. If you have one bone into the another bone, it should not getting fractured or anything. But the cartilage will give the protection onto that part. And ligaments, you have to move your hand or leg in a proper way. Otherwise, what will happen? You get a fracture or ligaments and you get a sprain to get on that. So you have to be very careful on the ligaments and tendons. They are supporting tissue, they are connecting tissue bone to bone or bone to muscle, and then you have a cartilage in between the bones. So all of them should be helping in protecting our body, our parts. So we also studied the types of fibers like elastic, collagen, and reticuloendothelial fibers. And also the collagen is made up of amino acid. What is the collagen? Collagen is a protein, right? It's a protein, and it is made up of amino acids, right? What type of amino acids? If you see the classification of amino acids, the percentage of amino acid, glycine present 34%, and proline is present 12%, and hydroxyproline is 10%. See, when in the synthesis of collagen, I'll give the collagen is synthesized in the fibroblast. This is the one fibroblast cells. And then here, the cells in the endoplasmic reticulum, where the protein synthesis is going on, rough endoplasmic reticulum, the ribosomes. You have the polypeptide chain of procollagen, which is being synthesized, and the transport into the vesicles, and then Golgi complex. Now the collagen, small little collagen molecules is being liberated outside. That's why it is called extracellular protein collagen. So the fibroblast now synthesize the collagen and pushing outside the cell. The outside the cell it polymerized here. And this site, when the procollagen, the glycine and proline rich collagen or procollagen, it will be hydroxylated into the proline molecule. So this is the active collagen molecule, hydroxyproline. So if you don't get hydroxyproline in your system, then that is become a you get a problem. There's a disease. The collagen is not being formed. So that's on several occasions, starting from arthritis and doing the vitamin C. Have you ever heard of vitamin C, like ascorbic acid? Ascorbic acid is necessary for converting the proline to hydroxyproline. The hydroxylation step involves ascorbic acid. If you don't eat vitamin C rich fruits and vegetables, what will happen? You won't get the hydroxyproline, you won't get correct collagen, and you will suffer in a scurvy. Have you ever heard of that, scurvy? The disease with a vitamin C deficiency. Okay, now this collagen is synthesized and make a bundle, and then fiber is formed, and these fibers and make a collagen bundle, and this is the collagen where we get it. So these are the cycle where the collagen is being synthesized from fibroblast and the synthesis of a final collagen step. So the origin of collagen, which is coming from, I'll write it the origins of collagen. Origins of collagen. Fibroblast produce collagen. Osteoblast produce collagen. That is present in bone. Okay. In fibroblast, it is in the tissue spaces. It produces collagen for tissue spaces. Osteoblast mainly for bone. And then chondroblast produces collagen. Chondroblast produces collagen, and that is mainly on cartilage. And then you have a smooth muscle that also produces collagen. Smooth muscle produce collagen that is present in blood vessels. Blood vessels or capillaries. Blood vessels and capillaries. Okay. And also organs. The collagen present in the organs, that is coming from the smooth muscle cells. So these are the cells, and these are the place where it export the collagen. So chondroblasts export collagen to cartilage, osteoblasts export collagen to bone tissue, and then fibroblasts, that's on tissue space, they export. Smooth muscle cell export the collagen synthesis and export to blood vessels and organ for their protection. Okay. These are the stages of collagen synthesis and where you find. Now, the fibers and ground substance which we extensively study, and some of them are the sulfated chondroitin 4-sulfate and different types of ground substance are there. These are the fibers, what the collagen is a type of fiber. Now I'm going to discuss about the ground substance. Ground substance. Ground substance, mainly we call it as a matrix. Like a bone cells present in a matrix, like a cement. If you build a house or a building, what you need to do, you have to make a cement, and then onto the cement you are going to keep your red bricks, right? The bricks, red bricks is there, right? And then these bricks, in between the cement will hold the bricks together. Is that right? The same way, the cement here in the connective tissue is we call it a matrix, and these are the cells, or the tissue cells. And this matrix, again, the ground substance, matrix otherwise called ground substance, and they have different types of either this ground substance as a liquid or solid or bone tissues or chondrocyte is more of chondrocyte. So it may vary to you. Okay. So some of these matrix, we call it the ground substance, they are sulfated, like a non-sulfated group. They call it as a chondroitin sulfate. I think we discussed earlier in our lab as well yesterday. Chondroitin sulfate. And they also have classified into two types. One is the sulfated and non-sulfated. I put a symbol for SO4 is sulfated chondroitin, and another type is the non-sulfated chondroitin. Here it is being removed. Non-sulfated, you get only chondroitin. CHONDROITIN, chondroitin sulfate. Chondroitin sulfate. There are different types. I'm not going to go in detail, but you have to remember that ground substance, they are made up of the chondroitin sulfate. And some of them are the liquid that we know about in the blood, right? And some other things are, these tissues is made up of white adipose tissues and then brown adipose tissues. Brown adipose tissues are mainly present in the fetus and the young animals, in the newborn. This one is the white adipose tissue in a scanning electron micrograph. See, look at this like a sponge. And these are the scanning electron microscope. And you get these capillaries which is going over. This is the capillaries. This one is capillaries going over there. And this is a reticular network which is there. Okay. And you will find the aerial nucleus you cannot see in this picture, but this is the scanning electron micrograph of the white adipose tissue. And the brown adipose tissue, where the brown fat cells, which will have, these are the mitochondria. The cristae of mitochondria more prominent, where the brown adipose tissue will produce heat in our body, in the young ones, and hibernating animals, because of excess of mitochondria. So that's another show for you. And you can also see these pictures in your figures, in your Blackboard supplement. And these are reticular fibers. How the reticular fibers are arranging in the cells over there. You can see that one. And the The next part is the macrophage system, or the cells, I mean, how the different types of the blood cells. We will see extensive on the tissue, blood, and other part of the connective tissue, muscles, and later on. But I'm not going to detail right now because we are going to see again, we are going to study again when we discuss about the blood of all those. But now you just get a feel of what we have studied in the connective tissues so far. Okay. So what I'm going to do, I'm going to review for your exam next week. Okay. Do you have any questions so far? You are going to see all the PowerPoints on chapter 1, 2, 3, and then prepare for your exam, as I mentioned. Okay. Where shall did you find any quizzes on the blackboard? Yes? Okay. For those who have not, I will just show you where to find. Okay. Into the blackboard. Are you in the blackboard now? Yes. You go into the assessment. Yeah, a little star over here. An assessment on this side. Okay, here assessment. Click that. You have practice quiz 1, practice quiz 2, practice quiz 3. Okay, and it is the end in September 16th. That is on the next week. Okay. All these three. I'll change this one. Availability we will extend to. Okay. So all of them. It is on September 16th.
Is that the day of the test?
Yes, that is right. Before the test, up to 10 o'clock, you have the option to watch. So you can go through this, but if you see this, save the answer, view the next, and then you can try all these questions. There are 33 questions for that particular chapter. I'm just randomly clicking on that one. I'm not going through the question each. If you say finish, and you will get. Sorry, just finish. Okay, I'm not answering this question. Okay, I'm just getting it. The quiz being submitted. View the attempt, and it might get whether I have a right or wrong. Just I want to view the attempt.
So the test is not next week?
This is next week. Next week, the same time, come here, and then I'll provide the computer and laptop, and then you are going to take the exam, and all these locations. Okay. So you have to practice that. You have all these ones. You will get only the score. If you get the score, yes, that is right. Sometimes the computer is tricky, so please check your PowerPoints to get the right answer. So in that way, it's okay. Also, you please watch in the whatever we discuss in the class notes which I have written on the overhead projector. Please read also for that, and you will get some short answer questions. Probably covered through my lecture as well as in the PowerPoint, some of them. Okay, so that's the part you have to prepare for your exam next week. Any questions from Victoria?
Dr. Soma,
Yes.
Are we working with the laptop computers for the test?
Yes,
or,
yes.
Or is it written?
You are there in Synchro Ranch. I will provide the computer for you, and you are going to take the exam in that one. Okay? And it's going to be a locked down browser or something. You have to do it. And during the class, so that's what we need to do. But is it okay in Victoria? You follow?
We're going to take it on Blackboard.
You have the computer in the classroom? Yes? Yes, you can take the exam onto multiple choice questions over there. And then I'll get the short answer questions which will cover all these 3 chapters. Do you have any questions here in Sugarland? Yes.
So we don't need a Scantron?
No.
No. This is going to be handwritten or?
No, it is the computer. You have to type your
So I need
answers.
to bring a computer?
No, we will provide you the
Oh,
computer. Yes.
Can I bring a mouse?
Yes. I don't know whether that's compatible or not, but yes. Yeah. You can do any attempt.
But those aren't graded, right?
No, no, it is not graded. It is not graded for your exam. No,
Just to
it is
practice.
just for practice. Because you are doing your homework already for the summary of all. So write your homework, and that will help you to prepare for the short answer questions too.
How many questions are on it?
You combine all these three, probably it's around 100 of questions.
100 multiple choice plus.
Some short answer questions. So the time is going to be 10 to 12. So be in time, and don't come after 10:15 or 10:30. I'm not going to allow you to sit for your exam. So be in time to take the exam. Okay. Any other questions?